Anatomy of eye lens. Anatomy of eye lens. Here we will discuss about the detailed anatomy of the eye lens. Then in the next lecture we will talk about the physiology of the eye lens. It is a transparent biconvex crystalline structure. Biconvex, transparent crystalline structure which has two diameters. The anterior diameter, which is 3.5 millimeter at birth and increases 0.02 millimeter per year. So this is the anterior diameter. This is the anterior surface. This is the posterior surface. Okay, so this is the anterior diameter. Now the equatorial diameter. That means these are the two poles, the anterior pole and the posterior. This is the equator. Okay, so this is the equatorial diameter, 6.5 millimeter at the birth, and it becomes 10 millimeter at puberty, and it remains constant. So it is suspended. It is suspended by suspensory ligament or zonules of Zinn through the ciliary process. With the ciliary process, these are suspended, and the zonules are more in the anterior curvature in both sides. The zonules are more in the anterior curvature. Now the radius of the curvature, the anterior and the posterior radius of curvature is 10 millimeter. It is 6 millimeter, and the anterior radius of curvature is 10 millimeter. It is present in a fossa, in a space known as patellar fossa, and beyond the posterior surface there is the vitreous humor. So between the vitreous humor and the lens, posterior surface of the lens, there is a retrolental space where it is attached with the vitreous by Weger's ligament. Now it has a capsule, anterior lens capsule, posterior lens capsule, and a cortex inside, the nucleus inside, and between the cortex and the anterior lens capsule, not posterior, anterior lens capsule, there is a anterior lens epithelium. Now it is the cortex, the fibers, the cortical fibers are made up of type 4 collagen. Now, this capsule is composed of a smooth homogeneous elastic envelope which has a thin part in the pole. So in the pole the capsule is thin. In the pole the capsules are thin. It is of 4 micrometer in diameter, but in the posterior preequatorial and the anterior preequatorial area the capsule is thick, 23 micrometer and 21 micrometer respectively. Now the anterior lens epithelium is a cuboidal cells which is present in the anterior lens capsule. It is metabolically active cell. So the power of the lens is plus 16 diopter to plus 19 diopter. It has no blood supply. So the nutrition it gains from the fluid of the vitreous cavity. Okay. Now the refractive index is 1.406 in the nucleus, that means here, and 1.386 in the cortex. It has a fetal nuclei. So embryonic nuclei is at the 3-month. The nucleus is present, embryonic nuclei. Beyond around the embryonic nuclei there will be a fetal nuclei, 3 to 8 months. Then it is surrounded by the infantile nuclei, then the adult nuclei. So the much aged nuclei is the embryonic nuclei. Embryonic nuclei is at the center, which is very much old nuclei. The fetal nuclei has a two-part, anterior suture and posterior suture, which is Y-shaped anterior suture and inverted Y-shaped posterior suture. Anterior suture is interdigitation of the apical cells process, and the posterior suture is the interdigitation of the basal cell process. So it is all about the anatomy of the lens.